Thanks, Natasha, for the nice introduction, and thank you, Stem Cells, for giving me the opportunity to present some of our work that we have done in our lab over the past year. I usually like to start my talk with some of the historical stories about chimerism. Then I will explain briefly the principle of chimerism in stem cell transplant. In the second half of the talk, I will focus on validation of cell lineage isolation for chimerism testing. And at the end, I will wrap up the presentation with some case studies. The word chimera came from the ancient Greek writer Homer's. Homer's described the chimera as a monster with the head of a lion, a body of a goat, and a snake for the tail. Although the chimera was killed in the story, we carried their name for the century. Another example of chimera is Baron Ashura, that appeared in the Japanese animation known as Messenger in 1970. Baron is one of the bad guys, and it was combined from mummies of a man and a woman. In biology, chimera is not as scary as in myth. Simply in human, it means a person contains cells from two or more different individuals. In 1960, a baby girl was born in Seattle Hospital. She was healthy, though one of her eyes was brown and the other was blue, and her organs were a bit unusual as well. On one side of her body she had an ovary, on the other side a testicle. When the doctor tested her blood, they found a mix of two blood types. The little girl in our example is called tetragametic chimera because 4 gamete cells came together to create her. 2 of her father's sperm fertilized 2 of her mother eggs, resulting in 2 embryos that merged into 1. Now, the fact that the 2 original embryos were of opposite sexes made this case of chimerism easy to see at the genetic level, but sometimes there is nothing obvious indicating a person might be a chimera. In 2002, a woman named Lydia Fairchild was divorced from her husband, and in order to qualify for social welfare, the authorities has to prove that these kids are her biological kids. DNA testing confirmed that she shared much less than a parent should, so authorities accused Lydia of kidnapping the kids. At that time, Lydia was on her 4th pregnancy, and there was a researcher who published a similar case in New England Journal of Medicine, learned about her case. The researcher talked to the judge and convinced them to take a sample from the infant after the delivery and examine their DNA. The judge then ordered a court officer to be present at birth. That officer later witnessed the neonate being delivered from Lydia's body and watched as doctor drew blood from both Lydia and the infant. After 2 weeks, the court received the result. The infant was matching the other kids, but not matching the mother. But then researchers looked at DNA from other different parts of Lydia's body that matched her children. Because of this result, the court allowed Lydia to keep her kids. Lydia was a chimera too, but in her case, 2 female embryos had fused in the womb. She was her own fraternal twin sister. Now, genetically, some parts of her body made her the kid's aunt, while the others made them her mother. Now, this sounds like 1 in a million case, but recent research shows that chimerism is far more common than what we thought. Singer-Taylor mole is another example of fraternal twin that was absorbed in the womb. The condition explains why Taylor has a large birthmark on her upper body. One side has a different skin pigmentation color than the other side, the result of her twin's DNA. Her chimerism has also led to an autoimmune condition because her body sees her twin DNA as foreign and reacts to it. She appeared in the doctor show and talked about her case. If you are interested, you can watch her whole story on YouTube by searching the doctor show. And chimera is not only restricted to human, but now we can see it in our cars. So if you are driving a hybrid car, then this is a chimera also. Now we will switch gear and talk about microchimerism. Microchimerism happens when the foreign DNA population accounts for less than 1 to 10% of the host cellular content. In 2012, a group of researchers examined the brain of 59 women who died mostly in their 70s. They were looking for a gene that was found only on the Y chromosome. They ended up finding the gene in almost two-thirds of the woman's brain. These women were chimera. They had cells with male DNA, and that male DNA most likely came from sons the woman had given birth 2 decades earlier. Later on, scientists learned that during pregnancy, some cells from the fetus enter to the maternal circulation and migrate to various organs. Some researchers suggested that microchimeras make you more prone to autoimmune disease. Another group of scientists suggested that the immune system kicks into the higher gear after giving birth, possibly to clear away leftover fetal cells. This defense may pose its own risk because women with autoimmune disorder such as rheumatoid arthritis can have relapse after pregnancy. And microchimerism could be natural or could be artificial. Example of natural microchimerism, which I have discussed earlier, are pregnancy, miscarriage, or twins. On the other hand, example of artificial microchimerism are organ, tissue transplant, and blood transfusion. Now let's focus on stem cell transplant and learn more about the principle of chimerism. At some points, healthy individual may develop a disease state like leukemia, seen as red cells in the cartoon. Then they receive some sort of treatment, and after treatment, they should have decreased in their immune system. Now the donor cells come along, have blue cells in the cartoon, and this donor replaces the immune system of the patient. If you have a complete chimerism, then the immune system is completely from the donor, and theoretically you should not have any further problem. But of course, this is real life. So you may have mixed chimerism where either the recipient regular cells come back, or in the worst case scenario, the cancer cells come back. So how we evaluate chimerism in post-transplant patient? This is done by taking a DNA sample from the post-transplant setting and compare it to the pre-transplant setting, which is known as the genomic sample. So in the next part of this presentation, I will talk about our lab validation for cell lineage for chimerism testing. In our lab, and in order to increase the sensitivity of our assay, we use cell enrichment method. So if you directly isolate DNA from the whole blood, this is called unfractionated DNA. If you isolated CD3 from the whole blood, then extracted DNA, then the DNA will be coming from the T cells only. If you isolated CD33, CD66 from the whole blood and extract DNA, then the DNA will be coming from the myeloid cells only. And manual cell isolation is not as simple as I just showed you in the previous slide. The process is very tedious and it takes almost 45 minutes to isolate one cell subset. So if two patients came in the same day, then this will be approximately 3 hours hands-on time. Sometimes we even get 3 patients in one single day. So the story started in 2021 when I met Michael at the ASHI meeting and I was complaining about the manual cell subsets isolation. Michael started to introduce the RoboSep to me and I was impressed by the amount of time that we can save. He even convinced me that by bringing RoboSep, it can reduce the hands-on time to only 5 minutes compared to 3 hours that I have previously shown you. And here is the picture of the RoboSep. The RoboSep is basically a liquid handler that can do automatic cell isolation for up to 4 cell subsets at the same time. And you can tell here that our tech already fell in love with it. Another procedure that I found that actually significantly reduced the turnaround, the hands-on time, was using this microscope. So in the past, we used to count the cells using the microscope and the hemocytometer. Then we calculate, then the calculator to calculate the concentration. This process takes almost 10 minutes to be completed per sample. To be more efficient, this is replaced now by the Countess cell counter, where you can get result in under a minute. From the screen, you can visualize the live and the dead cells. In addition, it can give you the final concentration of the white blood cells. Our initial inquiry was focused on determining the optimal collection tube. So we compared the EDTA as a collection tube and compared to the ACD tube. After testing both the EDTA and the ACD tube, it became evident that the ACD effectively preserves cells compared to the EDTA. The EDTA tubes resulted in approximately 25% dead cells, whereas cells collected in the ACD tube exhibited 100% viability. So in all our future experiment, we were focusing on ACD-only tubes to complete the validation. And here we did a comparative analysis of cell count between the manual technique and the RoboSep. The figure clearly illustrates that RoboSep consistently yielded a significantly higher absolute cell count in both myeloid and lymphoid lineage compared to the manual method. We next wanted to validate our lymphocyte isolation. So we used here the negative selection kit from stem cells. We then conducted a comparison of cell purity for the total lymphocyte between the manual cell isolation and the RoboSep using the flow cytometry. And examining the side versus forward scatter, it is evident that the purity of our lymphocyte was consistently above 88% for both methods, while the population isolated by the RoboSep exhibited a denser distribution due to higher yield. And if you like graphs, I added a graph representation here to confirm to you that the cell purity were almost identical between the RoboSep and the manual cell isolation. And for the T cells isolation, we use the CD3 positive selection kit. And to assess the purity, we use a bunch of different antibodies. We use the CD45PE, which label all the hematopoietic cells except the erythrocyte. Additionally, we use the CD2 and the CD5 was performed. So first, we gated on the CD45 cells so that we can isolate all or get on all the white blood cells. And then we took this population further to confirm that this population is consistently mainly of the our T lymphocyte. And here you can see we use the CD2 and the CD5 antibodies. The reason we didn't use the CD3 as an antibody because the positive selection used the CD3 antibodies for pulling up the T cells by the magnet. So now the binding site of the CD3 on the antigen has already been occupied. So in that case, we use a different antibodies to pull or to confirm the cell purity by using the CD2 and the CD5. And you can see that the population or the purity of our cells is almost 99.6% pure. And upon comparing the manual procedure to the RoboSep procedure, it became evident that the cell purities were nearly identical. I gated on the entire population to ensure that the cells outside the gate are not white blood cells, thus confirming their negligible impact on our downstream application. A comparison of the CD45 between the two figures revealed consistent cell purity. For those who prefer visual representation, I have graphed the result to highlight the closeness of the findings. Myeloid cells were isolated using the CD66 CD33 positive selection kit. Purity assessment involved staining the cells with CD45 PA, and furthermore, the cells were stained by CD14 and CD66, targeting the monocyte and the granulocyte respectively. After isolating the white blood cells with the CD45, gating on the CD66 and CD14 population demonstrated an impressive purity of nearly 99.6%, considering both monocyte and granulocyte. And upon comparing the manual procedure to the RoboSep procedure, it became evident that the cell purity were nearly identical. I also gated on the entire population to ensure that the cells outside the gate are not white blood cells, thus confirming their negligible impact on our downstream application. A comparison of the CD45 between the two figures revealed consistent cell purity. For those, again, who prefer the visual representation, I put them all in one graph to confirm that both are very close findings. Now, in the last part of this talk, I will focus on some of the case presentation. In our first case, which is a case of a subset chimerism, these actually are two different patients, patient 1 and patient 2. So if the physician is looking at only the unfractionated cells and see 100% in patient 1 or 99.4 in patient 2, and most of the lab will report 99.4 as 100% due to assay sensitivity, the physician will think that the patient are doing really well. But looking at the T cells in patient 2, it's actually a case of mixed chimerism, because patient 2 has 5.9% of host cells coming back. This can be indicative of or a start of a graft failure. In addition, looking at different cell subsets are of a significant value rather than just looking at the whole unfractionated blood. So case 1 is a clear case of a subset chimerism, which is defined as mixed chimerism in one cell type and the presence of recipient-only or donor-only derived cells in the other. This could be an early time point after engraftment or could be a start of relapse. In case 2, which is a microchimerism in cell subset, when you look at the percentage of donor cells and the host cells in the unfractionated sample of the post-transplant patient, the total must be 100. But in that case, it was only 99.6%. So where is the remaining 0.4%? Let's see the rest of the lineage to understand what's going on. In the myeloid lineage subset, the total is 100%. And all are donor. So the problem is not here. Now, when you look at the lymphoid lineage, you can see that the total is 97%. And again, there is a missing 3%. When we dug more into this patient, we knew that the patient was transplanted previously and had a failed transplant. When we added the DNA of his first donor to the analysis, we found that 3% originate from the failed transplant of the first donor. So let's see how can I explain this case much better. So conclusion of this case is actually a case of a microchimerism in the cell subset. As you see in the figure, microchimerism is the presence of small number of cells in an individual that have originated from another individual and are therefore genetically distinct. So this patient was transplanted by the red cells, and after the transplant failed, he received another transplant from the blue cells. Most of the red cells are gone, but few remains, which is known as microchimerism. And we already explained the significance of microchimerism at the beginning of this presentation. In case 3, which is a case of a split chimerism, in this case you can see that the patient has completely lost the myeloid lineage while remain some of their lymphoid lineage. And if you look at the graph trend over time, shows gradual loss of the myeloid lineage as seen as the orange line, and you can see that the lymphoid lineage seen in the blue line had barely changed over time. So this is actually a case of a split chimerism, which is a complete or mixed chimerism in one cell type and the presence of recipient-derived cells in the other. This patient has a recurrence of acute myeloid leukemia. That's why all the donor myeloid cells was replaced by the cancer cells again. In case 4, which actually explain a case of a primary graft failure, the patient started to lose their graft by day 64. So chimerism analysis can aid in the diagnosis of a case of a primary graft failure. And why primarily? Because none of the subset reach 100% donor chimerism and because of the short duration of engraftment. And this is another case of a secondary graft failure. So comparing it to the previous case, this is a secondary graft failure because the myeloid lineage reached 100%. And then the patient started to lose graft as shown where the red arrow is pointing. Case 6, this is a case of a mixed chimerism across all lineage and over the entire of the disease progress as shown in the graph. So mixed chimerism could be due to either the recipient regular cells come back or in the worst case scenario the cancer cells come back. And here is a disclaimer that chimerism cannot tell if the cancer cells are coming back or the healthy cells are coming back. And this is known as a limitation of chimerism. This can be done by MRD, which is a PCR done using specific marker that expressed only on cancer cells. And here in this case, or case number 7, this can show you that this patient was not doing very well until day 244, where he received donor lymphocyte infusion, leading to subsequent progression of complete donor chimerism. And this is the last case for this presentation, and I found this is one of the very interesting cases because this was a 45-year-old female. She was diagnosed with acute lymphoblastic T-cell leukemia. The patient was candidate for an autologous stem cell transplant. And what is autologous stem cell transplant? This means that the stem cells from the patient are taking, and then the patient are giving a conditional regimen. Then the patient is reinfused back with her own cells. By doing this, you can eliminate the graft versus host disease and the use of the immunosuppressive therapy. The physician wanted to try something else. The patient had an identical twin, so the bone marrow team decided to use her sister as a source of autologous cells. So in order to proceed with her identical sibling as a source of autologous transplant, they need to confirm that they are actually monozygotic twins. So they ask us for help. So first, what we did HLA typing to confirm first that they are matched at the HLA level. And as you know, HLA is not the only test that can conclude that these patients are a monozygotic twin. So we conducted chimerism testing, and because there was no informative marker between the donor and recipient, we concluded that this patient is most likely going to be an identical twin to her sister. So there is certain things that can affect the engraftment monitoring. Number one is the type of the disease and the donor characteristic, the intensity of the conditioning regimen, the sensitivity of different cell types to chemotherapy and radiation, the type of transplant, whether it's an umbilical cord or bone marrow or peripheral blood. Umbilical cord is much slower and peripheral blood is much faster. But again, peripheral blood, they have tendency to develop more graft versus host disease. And the dynamic changes in the chimerism level, immunosuppressive and donor lymphocyte infusion. And what is the limitation of chimerism testing? And I already mentioned that chimerism cannot detect between host cells and malignant cells, and you need another test which is called the minimal residual disease that can be done by PCR that detects certain marker present in the cancer cells. So a combination of chimerism testing with mixed minimal residual disease assay is the best way to detect relapse. And again, is the purity of your cell subset isolation. So in summary of using cell-specific lineage, it improves the sensitivity, the precision, the specificity of the assay. It can differentiate between subset chimerism, split chimerism, and mixed chimerism. It can differentiate between primary and secondary graft failure. It can predict graft versus host disease. It's important for monitoring of donor lymphocyte infusion, and it's important for early detection of relapse and detection of microchimerism. Before I end my presentation, I hope I had convinced you that these amazing people behind the scenes are doing all this work that improve the patient health and safety, and I'm only here to present their data. Thank you so much for listening.